L109. Then came the EU GMP. Then we have the ISO classifications. Now, what are the difference between these three different types of classification of clean rooms? For example, the differences between the various clean room standards are lie in their classification systems. Yeah, just like that. Lies in their classification systems to categorize clean rooms based on the maximum allowable particle sizes per volume of clean air. When I say classification system, when I say class 10,000 or class 1000 or grade A or grade B, it is all about the number of particle size which are permitted to be in a given volume of air. That is the basis or the premise of a classification system. Now, how does these three different types or the versions of the clean room differ is they have their particle size limits. When I say that, each the standards, the different standards may have variation in the size range of particles considered in the classification. For example, ISO 14644 considers particle down to 0.1 micron, while the older US Federal Standard 209E only considers particle up to 0.5 micron. So there is a difference in the maximum particle sizes which each of these classifications define. Means for US earlier it was 0.5 micron. Now ISO 14644, which is the mostly followed by all the pharmaceutical, take 0.1 micron as the minimum particle size. That's the one of the differences in the different classifications which we are talking about. Now, the other difference is the sampling method. The methods for sampling air particles can differ between standards. The number of this includes number of sampling points, the duration of sampling, and the statistical approach used to analyze the clean air also differs from standards to standard between US, EN, European standards and ISO standards. So these are the 2-3 differences which we have talked about. I will show you the table in which you can see that classification difference. But all of these standards are having the primary aim of achieving the clean environment within a given manufacturing space. But only there is a small difference in the type of number of particles allowable within a given volume of air. Seshadri, can we go to the next slide? Yeah. Comparison of standards. This is one of the slides which will give you important thing. If I look at the particles under US Federal Standard 209, which was the very earliest standard for classifying the clean room, when I say class 1000, it allows 1000 particles of particles greater than 0.5 micron meter per cubic feet of air. Now class 1000 is under that. Now, similarly, if we look at class 1000, which is under ISO 14644-2015, which means that ISO class 3, it is limiting the particle size to greater than or equal to 0.1 micron only, not 0.5 micron. So there is a difference in terms of the particle sizes. So therefore, when we are saying when we are saying that I am having a classification of clean room, you should be very clear under which standard it is. Normally, by generic thing, it is ISO 14644-12015, which we say ISO class 1, 2, 3, 4, 5, 6, 7 based on the particle sizes. And it is limited. And when we say, for example, class 5, class 6 is 1 lakh, that means that greater than 0.1 micron is 1 lakh particle per meter cube of air. Now comes the EU GMP. The EU GMP is having its own grades. That is, it doesn't call it as class 1, 10, 100, 1000. No, it calls it as grade A, grade B, grade C, grade D, and it has its own allowable particle per cubic feet. That is, say for example, grade D. If for example 0.5 micron, is 3 lakh 35 lakhs 20,000 particles, which is allowable. Similarly, if you look at in the same table, ISO 14644 for 3 lakh 35 lakhs 20,000, 0.5 micron, it is ISO class 7. So grade D in EU GMP corresponds to grade 8, sorry, grade 8 in ISO. So therefore, these are the ways of comparing, but normally we follow the ISO grades of classification of clean air. Now, another important thing which you should know is that usually the when the clean room is completed, everything is in a static position. Machineries, there will be no movement of people, no movement of material within the clean room. Then, when we check the particles using a sampling port or sampling, which Mr. Seshadri has explained yesterday, how do you do the sampling, checking, and other thing? It is usually at rest because there is no production activity which is going on, which is defined. But in when the production is happening, then it is called the clean room classification in operation. There will be a different particle counts which is allowed in the overall facility. In the number of particles which are allowed in the clean room facility. Usually, now no, there will be usually you will be dealing with aseptic. I'll come to that aseptic and sterile manufacturing difference. But ISO 14644 has largely replaced US Federal Standard 209E as the international standard for clean room classification and monitoring. While there are differences in particle size limits and classification schemes, efforts have been made to align the two standards. Now there is a conversion table which is available to to relate ISO classes to equivalent US federal standard classes. So as this table is very important for us to have an equivalency class between ISO to US federal 209E. Similarly, with the ISO 14644 versus EU GMP, that is the uh, European Union. ISO 14644 focus specifically on clean room classification and monitoring, while EU GMP covers broader aspects of pharmaceutical manufacturing, including facility design, equipment validation, and quality management system. So there is something which is EU GMP has gone a little further in terms of the standards what governs the clean rooms. Compliance to ISO 14644 is often considered as part of meeting EU GMP requirements, especially in areas related to clean room operations. So this, in effect, is the summary of these three standards which are available. You want to explain this? The tables which are provided to you in the slides will help you in understanding this particles and particle size, equivalency of those particles which I was referring to. Okay. Now there is one more point which I wanted to tell you is ISO 14644 only deals with particle contamination. But while you are going into the field and practically looking at it, ISO 14698 biocontamination control is also a new area which we have to focus on in terms of how to what the rules and the methods governing the biocontamination controls. ISO 14698. In practice, pharmaceutical manufacturers may need to comply with multiple standards simultaneously to meet regulatory requirements and ensure quality and safety of their products. Compliance of one standard often implies alignment with the others to some extent, but each standards like US, EU, as well as ISO has unique focus and requirements which we have to comply with based on the facility being compliant to the requirement of the facility. Okay, this is what is a summary of the various standards which I had to talk to you about. Now, the normally permitted activities based on EU GMP grades, if you read like this, grade A corresponds to, example I'm talking, grade A corresponds to ISO 5 at rest and in operation also. Grade B corresponds to ISO 5 at rest and ISO 7 in operation. Background support area for grade A zone. Like this, if you read the grade C corresponds to ISO 7 at rest and ISO 8 in operation. So this equivalency of various grades have to be known. One is that you should know the facility is going to manufacture as per which for which country, and what is the compliance in which it has to be, whether it is US FDA or EU GMP or PICS or ISO. Usually it is ISO which complies to most of the requirement, and there should be an equivalency established, and so that every aspect of the clean room environment is met. ಇನ್ನೊಂದಿದೆಯಾ? Grade D always corresponds to ISO 8 at rest. In operation, particle classification not predefined. Clean, controlled, unclassified, or clean, non-classified. These areas are called CNC areas. Now, just to give you a brief on qualification of clean room and clean air equipment, which all these various classifications dwell in, or how is the filter systems, the HEPA filter systems, leakage and integrity, airflow test, air pressure differential test, RH temperature, all these things will be captured in under all these classification requirements, so that it will be, if it is meeting that particular requirement, it will be classified as those clean areas. So, for example, class 8, class 7, class 5, or grade A, B, C, D. This table gives you a typical used air changes per hour of different grades. If you say ISO 8, which is equivalent to class 1 lakh, number of air changes is 24 to 50. And if I say ISO 6, it is equivalent to class 1000, and we have a number of air changes per hour as 150 to 200. So this was a summary of what I wanted to tell about the various classifications which are prevalent and how they are all interrelated to a greater extent. But importantly, they are all meeting the clean room environment requirement in terms of the air changes, temperature, humidity, or the particles which are allowed to be within the given space. Microbial counts, like for example, I said that new ISO 14698 controls the classifications of that. So this is in summary for this particular small topic of classifications. Now I open the forum for asking questions, which I know there will be many, but let me answer whatever I can. Sheshadri, you want to take some questions? Anything which the team wants to know from me on the various classifications? They are there, right? Silent again.
Sir,
Yeah.
sir, the percentage of HEPA filter coverage, that is 10 to 20% or what, sir? That for ISO 8, 10 to 20%. Like it
Yeah,
covers the area, whether the force will be higher in class, that is ISO 5, than the
correct.
ISO 8.
No, yeah, you are absolutely right. No, what you are meaning is 10 to 20% is the filter coverage area, is a approximation. For example,
Okay,
ISO 5, if you see the ceiling, the complete ceiling will be with the 80, when you say 80 to 100, the complete ceiling will be with the 80% coverage.
okay, sir.
And the number of air changes are 330 air changes per hour. If you remember
Okay,
yesterday's meeting, yesterday's thing, the pressure inside a very ISO 5 is very higher compared to the outside.
outside.
How do
Yes,
you achieve that? Is by the number of air changes, which are 330 or whatever number of changes based on the design. Correct? And
yes.
then to ensure that the laminarity, like what we discussed, the airflow pattern, it is that 80% of the ceiling is covered with that, so that uniformly the air is pumped in a vertical way and it is collected in the sides by the return air risers and goes back. And
Okay.
also, and what you asked me is a question which is the percentage coverage of the ceiling, which is 80%.
Okay, sir.
If you see a LAF, it is also class ISO 5 in a laminar airflow station. It is completely covered with the HEPA filter.
Yes.
All of that has a bearing. See, in any clean room, what is happening is you are only looking at the particle count, microbial count, and the number of air changes, temperature, humidity, because that is the environment which is basically defined by the various classifications, which will be meeting the production requirement for that particular pharmaceutical equipment or product. So that's where you have to be clear about it. Equivalency of the various classes, and whether it is EU GMP or whether it is ISO classification. But normally, when you say a clean room, it is classified as class ISO 8, 7, ISO 6, or ISO 5. And you should mentally remember that ISO 8 should belong, is equivalent to 1 lakh, ISO 7 is 10,000, like that. I hope I have answered the question.
Yes, sir.
Is there any other questions, or we can quickly shift to the next topic?
No question, sir.
Or we can assemble in another 5 minutes, and we can at 10:35 we assemble to start the aseptic versus sterile manufacturing. Okay.
Okay, sir. Okay. Thank you.
Hmm? ಎಲ್ಲಿದೆ ಇದು 1 minute, I'm just putting the slides. Can you see the screen?
Yes,
Difference
sir.
between sterile and aseptic
Yes,
manufacturing?
sir. Yes, sir.
Okay, fine. So now the last topic of this particular, my topic was on the aseptic and sterile manufacturing. Okay, so now what is the difference between aseptic and sterile manufacturing? See, the basic difference will be, let me go to the correct, no. The basic difference will be aseptic manufacturing involves preventing microbial contamination during the production of sterile products. Again, I repeat, aseptic manufacturing involves preventing microbial contamination during the production of sterile products by using clean room barriers like LAF or RABs, that is random access barrier systems, and sterilized equipments. That is aseptic manufacturing. Sterile manufacturing is a little bit ahead of aseptic manufacturing. Sterile manufacturing goes a step further by ensuring the complete absence of viable microorganisms through techniques like autoclave and sterile filtration. So there is a difference between aseptic and sterile. Sterile is the ultimate in terms of the clean rooms as well as microbial counts, whereas aseptic manufacturing facilitates the manufacturing of sterile products to be put in a under an environment, clean room environment. For example, I'll give you an example of what is aseptic manufacturing. Packaging of fruit juices in sterile cartons. For example, in the food industry, packaging of fruit juices in sterile cartons is an aseptic manufacturing. How? The juice is earlier is pasteurized to kill harmful bacteria, and then it is filled in sterilized cartons within a controlled aseptic environment to prevent contamination. That is aseptic manufacturing. Now, sterile manufacturing. What is an example of a sterile manufacturing? Surgical instruments. A day-to-day example is a surgical instruments. When you go to a OT, instruments used in surgeries are sterilized using techniques like autoclaving, which kills all microorganisms present on the instruments, ensuring they are sterile before use. Understand that? So these are the what is aseptic and what is sterile examples. Okay. Now, in terms of pharmaceuticals, if you look at it, production of pharmaceutical drugs in vials, that is a aseptic manufacturing. One when it is filled in vials in a under a laminar airflow or a RABs or what do you call that? Seshadri? Isolators. That is called aseptic manufacturing. Parenteral medication will also be manufactured under sterile process. Like for example, the parenteral drugs, which are the which goes directly into the body, are manufactured under sterile conditions to prevent the introduction of harmful microorganisms into the patient's bloodstream. That is also possible. Both aseptic and sterile can be done. Till now, I think I have been able to. It's a difficult thing. What is a very thin line between aseptic and sterile. So I'm just trying to give you some examples and making this topic a little lighter. Otherwise, it's a very little bit of a tricky thing. What is aseptic? What is sterile? Now again, I'll continue with that so that later on we'll take the questions. When to suggest aseptic manufacturing and when to suggest sterile manufacturing is a question which will come to all of you, correct? Now, aseptic manufacturing is typically suggested when it is important to maintain a clean environment during the production process to prevent contamination of final product. This is often the case for products like food and beverages, pharmaceuticals, and cosmetics. Aseptic techniques are used to reduce the risk of microbial contamination while handling and packaging these products. So, that is aseptic production. On the other hand, sterile manufacturing is recommended when absolute sterility is essential, such as for products intended for injection or implantation inside a body. This includes pharmaceuticals like injectable drugs, surgical instruments, and medical devices. Medical devices can be like, example is a stent, which we put in the heart, is also a sterile manufacturing. Sterile manufacturing ensures that the final product is free from viable microorganism, minimizing the risk of infection when used by patients or in medical procedures. So this is up to aseptic and sterile. I want you to whether you have understood that or not is a question which, because it's a difficult topic. So just ask questions if you have any in between. Hmm? So I suggested when to use aseptic, what is the difference? We have covered what is the difference between aseptic and sterile manufacturing. We took some examples and when to use aseptic and when to use sterile manufacturing. Now, there are different under this there are two three things called when we use the terms terminal sterilization. We use the term autoclaving. So these are the terms which we use as a sterile manufacturing process, wherein we are ultimately aiming to produce a product which are having zero microorganisms and it is filled in a cleaner environment. So I will talk to you about terminal sterilization. We had some terminal sterilization and aseptic area processing. What is sterilization? Terminal sterilization refers to the process of sterilizing a product, usually in its final packaging, to ensure that it is free from viable microorganism. Means the product when it is done, it is taken to to an equipment wherein it is heated or it is terminally sterilized so that any microorganism which is present will be removed. This is typically done using methods such as steam sterilization, gamma irradiation, or ethylene oxide gas sterilization. Terminal sterilization is commonly used for products that can, again, this is very important. Terminal sterilization is commonly used for products that can withstand the sterilization process and are not affected by it, because when you heat, the product should not decompose or change its composition. It is an effective way to ensure that the safety and sterility of medical devices, pharmaceutical, and certain food products are kept intact even if you are doing a terminal sterilization. Again, few examples. Terminal sterilization is generally used for simple solutions or small molecules like IV bag, water for injection, small molecule drugs not susceptible to heat in terms of the pharmaceutical thing. Again, one more point which you should note is the terminal sterilization controls the bioburden accumulation and then performs a final heat or irradiation sterilization stage after filling is completed. Means the product is complete, then it is going through the process as what I have explained earlier. In fact, the US FDA states that it is well accepted principle that sterile duct shall be manufactured using aseptic process only when terminal sterilization is not feasible. What does that mean is most of our vials, ampules, and injectables are all done under terminal sterilization because it is a sure shot way of saying that whatever drugs goes inside our body is free from any microorganisms or anything because of the terminal heating and sterilization process. Yeah, the intravenous or stent which we used for the or any replacement in the body with kneecaps and other thing. Now, then there is something called autoclaving. When is autoclaving suggested? So, autoclaving is suggested when sterilizing heating resistant material and equipment because certain materials are not suitable for terminal sterilization because they are heat resistant. Autoclaving is then suggested, such as surgical instruments, glassware, and media. You would have noticed in some when you go to QC that the glassware and media usually are autoclaved from one it is autoclaving is effective for items that can withstand high temperature and moisture without being damaged. How does autoclave work? Autoclave work using steam under pressure to kill microorganism, making it reliable method for achieving sterility in a wide range of applications, including laboratory research, healthcare settings, and pharmaceutical manufacturing. So this is where we talk about the difference between aseptic and sterile manufacturing, terminal sterilization process, and and uh, and autoclaving. Okay. Hmm? Hmm. Okay. Where is that sheet? Okay. Now, then, what is the area classification for? Uh, Australia. Hello, 11 and 20. That's still. Okay. Now, let me talk to you about what are the area classifications for carrying out aseptic manufacturing. That will give you a differential with respect to what are the area classification for sterile manufacturing. In aseptic manufacturing, areas are classified according to the risk levels of cleanliness and microbial contaminations. The classification is typically based on international standards like ISO 14644 for clean rooms and associated controlled environments, like what we talked in the first lecture. Usually this is the way it is done. ISO class 5, this is the cleanest classification and used for critical areas where the product is exposed to the environment, such as filling zones, vials, ampules. These are done under class 5. Equivalency of EU GMP is grade A. So that's where I had to refer to the earlier lectures. The air quality in this area must meet stringent requirement for particulate and microbial contaminations. ISO class 7 or grade B. The next stage after the clean area is the next cleaner area. This classification is suitable for less critical areas where product exposure is limited, such as buffer zones, preparation areas, because the final filling will be the grade A. The preparation areas, buffer zones will be in grade B, that is ISO class 7. Now, the outer areas are all under grade C or ISO class 8. This classification supports areas that are not directly involved in product handling, such as gowning corridors and rooms and corridors. The air quality standards are lower than ISO class 7. So always, if you look at EU GMP in an aseptic manufacturing, the filling or the product which is exposed will be in grade A, which is equivalent to ISO class 5. The next preparation to go into the filling area will be a little grade, lower grade, grade B or ISO 7. And then outside that, which are gowning and unclassified areas, corridors are all under ISO class 8, which is grade C, or sometimes it is grade D also. These classification help to ensure maintenance of appropriate cleanliness level in different areas of manufacturing facility to minimize the risk of contamination during aseptic processing. So this is how the aseptic processing is uh, concerned. What is the area classification for sterile manufacturing? Now that you have understood that aseptic manufacturing is having that, sterile manufacturing will also have certain grades of clean room areas. In sterile manufacturing, clean room classifications are crucial to maintain the necessary levels of clean rooms classifications are crucial to maintaining the necessary level of cleanliness and sterility throughout the production process. The classification system for clean rooms used in sterile manufacturing is based on ISO 14644. Now, how is the clean room done? As in the case of aseptic manufacturing, cleanest area where the product is exposed to the environment is ISO class 5, and then the surrounding areas will be in class 7 and class 8. But as what you have seen under sterile manufacturing, we are having terminal sterilizations and autoclaves, which will be under ISO 7 or ISO 5 or ISO 7, because that is the final product. When it is filled, it will be heated up to kill the microbials. Now, then it leads to, again, I've been referring to so many times that the vial and ampules are manufactured using this kind of manufacturing. So the question is, vial and ampule filling, is it a sterile or an aseptic manufacturing? Answer is, it can be done under aseptic manufacturing and it can also be done under sterile manufacturing. Vial and ampule filling can involve both sterile and aseptic manufacturing process depending on the specific requirement of the product, and important thing is the regulatory standards. In sterile manufacturing, vial and ampule filling process ensure the complete absence of viable microorganism in the final product. Again and again, I repeat, that's the only difference which is happening. That is done by terminal sterilization or autoclaving. But the product should be compatible for this. Otherwise, it will decompose. So hence, we have to take that as the primary differentiator between sterile and aseptic manufacturing. How is this typically involves using sterilized equipment such as filling machines and containers with clean room environment that meet stringent cleanliness standards. Sterile manufacturing is commonly used for products intended for injection or implantation into the body, where maintaining sterility is critical to ensure safety of the patient. Now, then it leads to the next question. When is aseptic manufacturing acceptable for vial and ampule filling? Aseptic manufacturing is often used for products that cannot be terminally sterilized, such as certain pharmaceuticals or biologics, or products where maintaining product quality is essential. Again, I repeat that both aseptic and manufacturing is complementing each other. Whereas in case of sterility, we are going for the final microbial killing using heat. Otherwise, it can be complementary and it can be used in based on the regulatory and the product specific requirements. So this is a simple description on aseptic and clean room and the classifications and uh, uh, what is the major difference between aseptic and uh, sterile manufacturing. I had only this much to talk about in terms of giving you a brief explanation of what is the topic which was given to me. Just to note, capital and operational costs are higher in case of aseptic processing because you use isolators, you use random access barrier sources to isolate and prevent any microbial coming into it. And because of that, the capital cost of HVAC systems are also much higher. Whenever a sterile product is capable of being made to the required regulatory standard with terminal sterilization, it becomes more viable to follow that method. So this is the summary of the overall lecture between aseptic and sterile manufacturing. Please let me know if you have some questions on this. Otherwise, I'll ask you some questions on what is the difference between aseptic and. Can anyone tell me one major difference between aseptic and sterile, which I've told you now?
So in sterile manufacturing, it is generally done at the after filling. It is after filling after filling of the drug product, it is sterilized at the last. Whereas in aseptic, it is by controlling the environment through the monitoring by using different techniques such as using isolators and wraps and other environmental
Correct.
monitoring. Aseptic techniques is done.
Correct, correct. So, but then it also depends on the regulatory requirements and also depends on the product, correct? But also one should note that in aseptic, the cost of manufacturing is higher because of the equipment cost of isolators, wraps and other things. Whereas in sterile manufacturing, once you fill it, you go to a terminal sterilizer, and because it is heat driven like autoclaving and terminal sterilizers, you don't have that much extra cost for maintaining the HVAC and other things. That's a point which you should remember. One question was there, so he is correct in asking it. So I think with that, if anyone no one has got a question, I think I can conclude this session. And with that, I leave it to Mr. Ramaya, right? Vasant,
Okay.
to close these sessions.
Hello.
Thank you so much, sir. Thank you for your time. If
Yeah.
any of you doesn't have any question, like later on you can contact any of the faculty. You have their contact details.
Yes, yes, yes. Please.
And on behalf of Biocon Academy and Ramaya College of Arts Science and Commerce, I wish all the students all the very best for their exam. And
Thank you, sir.
yes,
All the best for your exams and hope to see you
Thank you,
soon
sir.
in some of the industries which we are working, and you will be placed properly, and impart the whatever the knowledge you got will be imparted in the field. Okay, all the best.
Thank you, sir.
Thank you,
thank you
sir.
all.
sir.
Thank
Thank
you
you,
so
sir.
much.
Thank
Thank
you,
you.
sir.
Thank you,
Yes, sir.
Dr. Vasan. Thank you.
Thank you, sir.